तो चलिए क्लास, अब हम इस चैप्टर की समरी को देख लेते हैं। The enzymes that cut DNA at specific sites are called restriction enzymes. The specific site is called recognition sequence. The first restriction endonuclease was HIND2. More than 900 restriction enzymes have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences. Restriction enzymes belong to a larger class of enzymes called nucleases. These are two kinds, exonucleases and endonucleases. क्लास, अब आप वीडियो को 2 मिनट पॉज करिए और इन सब के नोट्स बना लीजिए। Exonucleases remove nucleotides from the ends of the DNA. Endonucleases make cuts at specific positions within the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequences in the DNA. Fragments of foreign DNA can be separated by a technique known as gel electrophoresis. The separated DNA fragments can be visualized after staining them with ethidium bromide followed by exposure to UV radiation. क्लास, अब आप वीडियो को फिर से 2 मिनट पॉज करिए और इन सब के नोट्स बना लीजिए। Vectors help in easy linking of foreign DNA and selection of recombinants from non-recombinants. An ideal cloning vector must have origin of replication, selectable marker, and cloning sites. In order to force bacteria to take up the plasmid, the bacterial cells are treated with calcium, followed by heat treatment and placing them on ice. Microinjection and gene guns are also used as methods of transformation. क्लास, अब आप वीडियो को फिर से 2 मिनट पॉज करिए और इन सब के नोट्स बना लीजिए। क्लास, ये कुछ रेफरेंस क्वेश्चंस हैं इस चैप्टर के। एनसीईआरटी का क्वेश्चन 7, 8, 10th का ए पार्ट, 11th का बी पार्ट और क्वेश्चन 12। वर्कबुक के क्वेश्चन 3, 8, 9 और 10।